Thank you very much for attending my talk today. I probably have the most difficult name, so sorry for the challenge for pronouncing my name. So I'm Agapitos Patakas. I'm the CSO of Antibody Analytics, and today I will present you our IndEx-2 platform, which is a cell line system to allow the assessment of the impact of antigen density on the efficacy and safety of immune-directed therapies, but not also ADCs as well, to a degree. Before I go to the main part of the talk, I would like to introduce you Antibody Analytics. We are an immunology-focused CRO. We are founded in 2015, and we're based in Scotland, just outside Glasgow. And this is one extremely rare picture of Glasgow without any clouds or any rain. We employ probably around 90 employees today, and we're most of them are actually experts in human immunology and bioanalysis of antibody-based therapeutics. We apply a unique combination of cell line development, molecular biology, immunology, and asset development expertise to develop quite advanced experimental models to address different questions that our customers have. And one of these models will be obviously the IndEx-2 system that I will present today. Well, obviously, as the name of the company suggests, we are focused on antibodies, but we also have quite extensive expertise in other advanced therapeutics, mainly in cell therapies, immune cell therapy specifically. We have a global customer base. As well, most of our clients actually are from the United States, and our expertise are actually applicable from very early discovery phases to even the clinic, especially with development of GMP ready, we call them potency assays for product characterization. So going back to the scientific part of my talk, there's not much I suppose, background required for this audience for this, but especially for immune engaging and immune activating therapeutics, but also other molecules like ADCs. One of the key challenges is actually identifying a truly tumor-specific antigen. In most cases, the antigens that are currently targeted are usually overexpressed in cancers, but they also will have some level of expression in healthy cells. But even if we identify a truly cancer-specific antigen, in most cases there is quite a significant heterogeneity in expression, either induced by the therapy itself or natural, that can result in development of resistance due to low expression of the antigen. In the case of obviously expression by healthy tissues, one of the main, I suppose, considerations is the on-target of tumor toxicity, and obviously previous speakers also suggested that this can be even stop clinical trials quite early due to this undesired side effects. And especially for CAR-Ts, there's a requirement by the regulator for assessment of these types of side effects early in the preclinical development of these modalities. And the most conventional approach, especially when functional data are required, is to, and the one actually that is suggested by the regulators, by the guidelines, is to employ a panel of cancer cell lines which usually express different levels of the antigen of interest, and employ them in functional assays. Obviously, the type of readout will depend on the mode of action of the therapeutic. And what is usually expected, that is the response, whether it's cytotoxicity, proliferation, cytokine production, will be proportional to a degree or correlated to a degree to the receptor number. There are a few limitations with this approach. First, I suppose it's a logistical issue. To develop a quite large panel of cancer cell lines can be difficult. A lot of the cell lines are in academic labs, and the access can be limiting. And there's cost, especially licensing costs, are associated in many cases that can make, I suppose, a relevant big panel quite a costly endeavor. More importantly, there are some methodological issues. The expression range, and I'll show you some data later, even from quite large panels can be quite limiting and might not cover the full physiological and pathophysiological range of expression required to cover that. And especially if things like threshold, determination of threshold of activation are required, this can also be limiting. However, the cell line background is actually can be confounding in the type of data that are generated, because in a lot of cases we assume that all the cell lines are identical apart from the level of expression of a specific target antigen. But in reality, they have inherent differences that can render them either susceptible or resistant to a specific type of therapy in a manner independent to the antigen expression. And if it moves, yeah. And this is actually a publication that quite nicely demonstrates this limitation of this approach. This is a publication for a bit old now, from 2015 by you and colleagues. And they were actually aiming to develop CAR-T therapeutics against HER2 and EGFR, which are very susceptible to on-target of tumor side effects. And they tried to obviously tune the activity of these CAR-Ts by changing the affinity of the antigen binding moieties of these CAR-Ts. And if we focus on the HER2 part of this study, you'll see the authors employed a panel of 11 different cancer cell lines. But even in the relatively extensive panel of the cell lines, you can see there's only 3 levels of antigens that can be, I suppose, observed: a high, a medium, and a low. And obviously, they employed also a negative cell line, which is the MDA468. However, when they applied these cell lines in functional assays, we got some quite interesting results. So there were 4 different CARs that were generated. All of them were based on trastuzumab, with 4D5 being the highest affinity and 4D53 being the lowest affinity. But if you see, when they co-culture them with the cell lines of different HER2 expression, you see there's no really close correlation between the level of HER2 expression and the biological activity induced after activation of the CAR-Ts, in this case, interferon gamma production. And if you focus, for example, on 4D57, you see very little expression of interferon gamma in high-expressing cell lines like SCOP3, and quite high expression in lower expressing cell lines like PC3s. However, when the author employed a common genetic background for their assays, so they used K562s, and they induced differential levels of expression of HER2 by transducing different levels of HER2 mRNA. The response was actually quite different. So in any, I suppose, affinity of CAR-T, you could see a clear correlation between the biological activity, in this case it's proliferation, and the levels of HER2 expression. So obviously, the higher, against the highest expressing K562s, there was the highest response. Obviously, they found that the second affinity was the one that was higher biological activity, but in its affinity, the correlation was clear. So what would be the ideal system then? Obviously, as I mentioned, the common genetic background is critical. A large dynamic range of expression is also very important, from a few hundred receptors to a few 100,000 receptors, so we can cover the whole physiological and pathophysiological range of expression. And also for us, it's important to be finely titratable, because in that way we can actually define thresholds of activation that is very difficult to do if you only have a couple of levels of expression. And obviously the availability of a negative control that you can benchmark any data you have against that. I believe we have a solution, or we have developed a solution that is superior to the conventional approach with the cell lines. We call that the IndEx-2 system from inducible expression. And this is a system that actually has 3 main pillars. It's an inducible, it's a cell line system that allows the inducible expression of antigens from a few hundred receptors to a few 100,000 receptors. And not only one antigen, actually, we can do that with 2 different antigens in an independent fashion. We couple that with a set of functional assays. We employ quite well characterized donor pools, and couple that with assays with multiple readouts. Obviously, the type of readout will reflect the mode of action of the therapeutic. And we couple that with an analysis approach that allows determination of thresholds in the cohort of either healthy donors or patient, depending on the type of questions that this need to be answered. So the cell line system that I mentioned before, the IndEx-2 system, utilizes the chemical proximity approach to induce antigen expression in a very titratable fashion. Actually, two antigens, and I'll show you examples of that. In chemical proximity systems, employ small molecules to induce dimerization of protein partners. They are very well, I suppose, converse systems, very important for cell signaling. If we pair one of the protein partners with a DNA binding domain and the other partner with a transactivation domain, then we can generate, as we call it, a molecular switch that we will pair with the gene of interest that is with an upstream activation sequence that is recognized by the DNA binding domain. We can induce the expression of the protein of interest in a manner that is highly correlated with the concentration of the small molecule. I suppose the most famous, probably the most applied chemical proximity system is the rapamycin-based systems. They have been applied in both therapeutic development and also for experimental models. But the ones we actually chose are based on plant hormones. Most of the data I will show you today is with abscisic acid and indole-3 acetic acid. There's a few reasons we selected them. First of all, they allow a highly titratable and expression with a very large dynamic range. There's quite very little background in the absence of the small molecule. But more importantly, both of the acetic acid and indole-acetic acid are quite inert molecules. They are plant hormones. We have very limited biological activity in mammalian cells. And this allows quite a lot of flexibility, especially when we try to develop experimental models using these systems. And this is the type of data we generate. So what is usually as a background cell line, we use the CHO cell line. And this is actually a convenient cell line. It actually acts as a blank canvas, as most human targeting therapeutics don't bind proteins expressed by the cell line. But in the case that we wanted to use a different cell line background, or there is actually binding, we can CRISPR out any unwanted protein as well. We have generated a portfolio of what we call inducible platform cell lines that incorporate one or more of the induction cassettes. And that allows us to quite rapidly generate inducible cell lines for the antigens we want to work as well. And this is just a few examples of the cell lines we have generated so far. This PD-L1, HER2, BCMA, and CD19. And we usually do the quantification of the receptor expression by using quantitative flow cytometry. So for terms of simplicity, we call that receptors, but in reality, this is antibodies bound per cell. As you can see, we can have expression from a few hundred receptors to a few hundred thousands, in some cases, million receptors. And there is a bit of variability between proteins as well. We can also do that two antigens at the same time in a single cell line, and we can control that in an independent manner. So this is, in this case, CD22 is controlled by the abscisic acid and CD19 by indole-acetic acid. So in the next few slides, I will demonstrate with a case study how we use the system. And this is work we did with a long-term customer and partner of us, Kalina Oncology, and that they are developing a T-cell engager against CD19. And CD19 is quite interesting antigen. Obviously, it's one of that are actually truly non-cancer specific. It's expressed by healthy B cells from very early development to obviously different types of mature B cells, but also is expressed by different types of malignancies in leukemia, lymphoma. And because of that, and also because of the fact that usually patients can survive with relatively limited clinical intervention without B cells, many therapeutics have been targeting CD19, and a few of them have been approved. Blincyto, the first T-cell engager, obviously Kymriah and Yescarta, the CAR-Ts, and obviously the notable is an ADC. However, even though these therapeutics are very successful, a proportion of the patients that are treated with them do not respond or relapse to after treatment with these therapeutics. And one of the reasons that this happens has been linked to the levels of CD19 expression. In this publication by Spiegel and colleagues, they actually tried to define this, I suppose, threshold of CD19 expression below which patients are, have higher chance to develop resistance to therapy or have a progressive disease. And this is specifically for CD19 CAR therapeutics for lymphoma specifically. And they used, again, quantitative flow cytometry to assess that. And what they found is in that patients that express around 3,000 or below CD19 receptors per cell have a higher chance of developing progressive disease. This is not only the case on lymphoma and CAR-Ts, this is also the case for CD19 T-cell engagers from blinatumomab, with a couple of studies also suggesting that this is one of the modes of development resistance. So it would be beneficial for a CD19 targeted therapeutic to retain its efficacy even at low levels of CD19 expression to reduce the possibility of developing resistance to CD19 downregulation. And this is actually what Kallinan, the scientists at Kallinan, tried to develop. So they're developing CLN-978, which is a very highly potent CD19 targeting T-cell engager. It has a very high affinity for CD19 and intermediate affinity for CD3. It also incorporates an HSA binding domain that denotes extended serum half-life. We have worked quite a lot with this molecule, so especially with target cell lines like Radis and Ramos that express around 10,000 CD19 molecules per cell. We can see a very high potency in terms of toxicity, T-cell activation by CD25 upregulation, and cytokine production like interferon gamma and TNF. But because of this high affinity against CD19, it's also predicted to function at low levels of CD19 expression. And we'll try to test that using our inducible cell line. So we induced several levels of expression of CD19 from, and I'll demonstrate from a few hundred receptors to a few 100,000 receptors. And we co-cultured them with isolated T cells. So it's important to do T cells in this case because obviously the endogenous B cells will express CD19 that can confound the results. And we applied two concentrations of the CLN-978. Both of them were related with their clinical relevant concentrations for the phase 1 clinical trial. And we assessed T-cell activation by CD25 upregulation, interferon gamma production, and target cell cytolysis using the Excelsen's real-time cell analyzer. And what we found is that as for all readouts we assessed, that CLN-978 retains its functionality even at levels of CD19 of a few hundred receptors. We can actually, just to be clear here, as you can see in the x-axis is the number of receptors, whereas the y-axis is the biological activity. And we can actually quite effectively quantify the expression levels up to probably for some 400 receptors, with the last three being more extrapolations as well. So just have that in mind. So as you can see, there is, especially in the high concentration levels, we have retention of the activity even in the lowest levels of receptor expression, both which is the same for T-cell activation and interferon gamma production. We want to push a bit the system further, so we want to see if we can use this system to also compare to different molecules. We see this as a tool, especially for early lead identification and lead selection. And also we also wanted to see if we can use the same system to define a threshold activation for a specific cohort. In this case, we use a cohort of healthy donors. We compared in this experiment CLN-978 against, I suppose, the typical CD19 engager with this blinatumomab. Again, the setup was very similar. We induced 7 levels of expression of CD19, and we isolated T cells from 10 donors and did the co-culture. And the main readers in this case were interferon gamma production and target cell cytolysis. And what we can see here clearly is that CLN-978, it actually retains the efficacy and actually is much more potent than the inside, at least at this concentration. I forgot to mention this is 225 picomolar, which is the lowest concentration from the first experiment I saw it. And it can retain its activity even at extremely low levels of CD19 expression to the 100 of receptors. This is both in terms of toxicity and the interferon gamma production, which again confirms what we saw in the previous experiment as well. So then we tried to move on and try to define a threshold of activation for the cohort of 10 donors. In order to do that, we used a statistical analysis, but it's called receiver operating characteristic analysis. This is actually quite widely used for diagnostic tool development. And in order to do that, we classified as responders, respond any biological activity, either cytolysis or interferon gamma production, that was 3 standard deviations above the response we have against the wild type choice, which is obviously don't express any CD19. And by doing that, we managed to generate these ROC curves. And by using a metric of the ROC curve, it's called the Uden index, which is basically the maximum distance between from the true positive rate and the false positive rate, we define the threshold of activation. And for CLN-978, we identified that was actually the 81 receptors, which again, take that with a pinch of salt because there's a bit of extrapolation on the number of receptors. But we found that for both cytolysis and interferon gamma, that was the lowest number of receptors we can detect. However, in the case of blinatumomab, there was difference between the two readouts. For cytolysis, it was around 850 receptors, but for interferon gamma production was much higher, around 9,000 receptors for this specific cohort of healthy donors. So if we go back to this threshold, we kind of defined of around 3,000 receptors. We can see that, I suppose, both molecules do push in terms of toxicity this threshold to around 800 receptors. But if we collectively assess the result, we can, I suppose, speculate that CLN-978 will probably have patients that were treated with this molecule will have probably lower risk of developing resistance, at least due to downregulation of CD19. This molecule is actually now in phase 1 clinical trial, so we're really looking forward to see if this result also will translate it in the clinic as well. Obviously, if the target was different, if it was, for example, a solid tumor, then the selection, I suppose, or the comparison will be, and the interpretation of the data will be very different, and especially with more advanced immunotherapies that want to unlink different types of readers, for example, cytolysis or interferon gamma production. This type of data can be quite useful for selecting the best lead or the best candidate for moving the clinic. So I hope that I have kind of convinced you that our IndEx-2 system is superior in terms of flexibility and quality of data generated. We believe that it's the only in vitro system that allows estimation of antigen thresholds for initiation of effector function of a therapeutic. And because of that, we think that it is quite suitable for hazard assessment of on-target of tumor side effects, especially for solid tumor therapeutics as well. It's applicable, obviously, for multiple types of therapeutics. I already mentioned with T-cell engagers. We have generated data with CAR-Ts, and this is just some example data with a bispecific CAR-T against CD19 and CD22, excuse me. We also have data with ADCs, and this is just some example data with trastuzumab-based ADCs with 2 different payloads. We have got some interesting data, especially in the higher levels of expression of HER2, and also conventional monoclonal antibodies. This is just ADCC data with different HER2 targeting therapeutic trastuzumab, and a combination of. So as a service, obviously, we would love to hear from you. Different types of therapeutics will have different requirements, different mode of actions. We're able also to help you analyze the expression pattern, the relevant expression that they required. We have access to lots of different cell lines and tissues, either from patients or obviously healthy donors. And obviously, we can couple that with the generation of cell lines that allow inducible expression. We already have a quite extensive panel of the cell lines, and we're expanding as we speak, actually. And we can couple that with functional assays, again, with multiple readouts that can obviously correlate with the mode of action of the therapeutic. And I hope I convinced you that we also have an analysis approach that allows determination of threshold of activation for specific cohorts of either healthy donors, that we did in this case, but also we can do it with patient material as well. So I would like to thank the antibody analytics team, the research and development team that developed that in the last couple of years, but obviously Kalina Oncology that was the first adopter of the technology and we allow us to use their molecule to exemplify a lot of the work I showed you today. We also have two posters, posters 25 and 26. Team is actually developed a lot of the cell line systems, so if you want more details about those, please go and talk to him. And Robert has done a lot of the immunology-based assays, and a lot of further other systems that we have developed to assess different types of therapeutics. Obviously, it's our commercial team, so as well, if you want to download this talk, please scan this QR code and you can have the full talk. Thank you very much.
Thank you very much. Thank you. Question?
Hi, I have twofold questions. One is this titrated expression system. Have you compared it to, for example, doxycycline-based systems and see how they compare? And that's question one. And question two is, when what's the maximum and versus minimum fold increases, like the range of dynamics that you can titrate it?
So, obviously, relative with the tamoxifen systems, well, there's two reasons why we didn't use because the small molecules are a bit more bioactive compared to the abscisic acid and indole-acetic acids. But we see a bit more leakiness in those systems compared to the ones we use. So you see expression in the absence of any inducer, which creates a bit of a background from what we see. So that's why the reason we selected those systems. In terms of dynamic range of expression, and so for most of them, we see from a few hundred receptors to up to 1 million receptors. So there is a huge range of expression, so we can get quite a big dynamic range.
So I have a question. So you look at the CD19, right? Yeah, particularly, and it build up different kind of density. So what kind of minimum the CD19 receptor per cell in your experiment you see compare ADC, for example, versus CAR T cells? You start to see the significant killing.
So with ADCs, actually, again, some of the molecules we have might not be of the greatest quality because we don't have access to molecules like that. We've seen that there are higher receptor requirements. So, for example, for the trastuzumab-based, we see a few, to see very high killing, more than like 100,000 receptors. But with CAR T for HER2, for example, we just have, I don't have any data to show here, but we just generated, we can see from like hundreds of receptors. So there is quite a difference between modalities. And I suppose it's expected as well.
Yeah, so that's probably expected. Yeah. One is like 10 to the 5th size per cell. The other you could kill as low as like 100.
Yeah, exactly. Yeah.
Okay, that's very interesting. And how about the CD22 or CD20? Did you look at that?
For CD22, we only have a dual CAR, so we didn't see the single one. And you can see from quite low levels of receptors that you can see killing. That was actually, again, from hundreds of receptors, not thousands, actually, you can see. But that was a specific CAR, and it was an OR CAR, so you can either work with CD19 or 22.
So it's also in 100 level, kind of.
Yeah.
Right. Okay, very good.